before we went on break, we had finished with nucleic acids. We finished with the structure of nucleotides, structure of the nucleic acids, and then looked at functions of the nucleic acids. That is gene expression, gene regulation. I think that's where we ended. Today we are continuing with the structure and function of hemoglobin. So a bit of historical background. Blood has been known to carry oxygen over a long time. I mean, since say the 18th century. That is like say 1793 thereabouts. I mean, some archives have shown that persons like William Withering, their letters they wrote. So William Withering was an English physician, a chemist, botanist. Let's just say he was a scientist who actually has been known for his works on the discovery of the active ingredient of the plant digitalis species, because he himself, as a doctor, has been treating some patients with some heart conditions, and one of his patients used a herbal product which worked so well for that patient. So he was very much interested in knowing what really was in that herbal mixture that helped the patient. So he was studying, that herbal mixture, wanting to know the active ingredient in it, and eventually found out that there was the plant's digitalis species, that contained the active ingredient, some glycosides, which is, some conjugants of carbohydrates and steroids that helped in the treatment of that patient. And so from then, it was upon his work that, some drugs like digoxin, which is used to treat various heart diseases, has been developed. And out of it, now many, medications for cardiac conditions based on some, the glycosides have been developed up to today. And in his communications with some of his colleagues, he referred to, what is referred to as factitious air, that he noticed, what he noticed with the interaction of factitious air with blood. Factitious air, actually, in those times, they refer to artificial air as factitious air. So that is, if you see factitious air, it only means artificial air. It's just like how today we have, say, oxygen, like you have an oxygen tank in the clinic that is being used, things like that. That is artificial air. So he was referring to how administration of, you can see here that he referred to factitious oxygen air that was administered. And he documented his observations of, the effect of such factitious air. So some, scientists, working on the subject of, blood carrying oxygen, sort of, chanced on some of these archives and then further worked on until, today. Another person, Thomas Beddoes, he too was a British physician. He was also a doctor. He basically was working on tuberculosis, pneumatic conditions. So, those respiratory tract conditions. And he also referred, but at the same time, he was a prolific writer. He published a lot of works. Apart from science, he even wrote on different things about nation building, politics, and things like that. But in those his scientific tests, he too referred to, factitious air. So some of those archives served as a background for, uh, modern studies on the property of blood carrying oxygen. Now, fast forward, you have a German scientist who, for the first time, so that is Otto Funk, for the first time in 1851, was able to crystallize the hemoglobin molecule. So from blood, he was able to mix blood with different solvents such as water, alcohol, petroleum ether, and from that mixture, he was able to carefully, with slow evaporation, have a crystallization of hemoglobin. So for the first time, that was a breakthrough to be able to do such a separation. You separate the hemoglobin molecule from blood. Because once you are able to do that separation, it means you can do further studies on that molecule. But of course, let's say it was extraction of proteins. It may not only be just hemoglobin that will be in the extract that he got, because there will be some other proteins in there. But let's say hemoglobin was of a major quantity in the extract that he got. So that was an improvement from the earlier studies. Then fast forward into the 20th century, modern times, that already this time, even though there has been methods or the thought of, say, light diffraction being used to study the structure of molecules, it wasn't until, say, the beginning of the 20th century, that is the 1900s, that the method of X-ray crystallography was, much developed, that it can be used to study the structure of molecules. And so Max Ferdinand, who is also an English, but he was born in Austria, and so he was working in a University of Vienna. But at the same time, he was also working in some university in England as well. And he was able to use the X-ray crystallography to determine the structure of hemoglobin. So that is big. Now you know the structure of hemoglobin. So he shared a Nobel Prize in chemistry in 1962 with John Kendrew, another Englishman, also a crystallographer. The two shared, that Nobel Prize in chemistry because Kendrew at the time also studied the structure of myoglobin. As we continue, you come to see myoglobin as well, so you can see the comparison of myoglobin with hemoglobin. But because of, these structures, they are proteins that contain heme. And so the Nobel Prize they share was like Nobel Prize in chemistry for their work on the structure of heme-containing proteins. So as we continue, you come to see what a heme is, and you relate with why it's referred to as heme-containing proteins. So we say hemoproteins. So structure of hemoglobin and then the structure of myoglobin were determined at this time. And there was that Nobel Prize that was given to these two. I just think that sometimes when we study some of these things, we should get to know some history behind it. So we begin to appreciate that, people really worked in the past, did so many things, even with, little resources. They tried their best to, lay some foundations. And as time went on, others also, you know, uh, built on the information. Before today, we younger generations, we are studying these things. So we can appreciate the work of some of these pioneers in the field. Now, before we continue, I'd like you to bring your mind to the fact that you can categorize proteins into two major classes. That is globular proteins and fibrous proteins. Because you hear in the name hemoglobin, you hear globin. The globin refers to the fact that that protein is a globular protein. And the first part of the name, hemo. So heme, that is a prosthetic part of the protein. So I know in the first year, when you did biological chemistry, you would have studied, maybe you talked about enzymes, for instance, which are biological catalysts. And these enzymes act with what we refer to as cofactors, which are metal ions, like copper ions, magnesium ions, ferrous ions, etc. And these metals, though they are non-protein, they are required for the functional protein structure. So they are there as cofactors. They are required for the protein to be functional as an enzyme. And so we refer to it as a prosthetic part. Sometimes we say an allosteric part of the protein. So that is the heme. In the case of hemoglobin, we'll come to see it clearly when we are moving on to look at the structure of hemoglobin. Because the fibrous proteins, those are, fibrous. They are for structural proteins like the structural proteins you find in muscle cells, for instance, or you refer to proteins such as fibrin, which is responsible for forming a sieve over, a cut skin, a wounded skin, that to trap blood cells in order to form, blood clots to stop blood from, oozing out of the wound. So to stop bleeding, you find proteins such as fibrinogen convert into fibrin to form that mesh for healing. So that is a fibrous protein. But hemoglobin is a globular protein. That's what you find in the name as globin. Right. Now, you want to remind yourself of some basic things which you already know. That hemoglobin is the pigment that is responsible for the color of blood. That's red pigment is coming from hemoglobin. And that you find in which of the blood cells, in the red blood cells, what you also call erythrocytes. The concentration of hemoglobin in males and females, the difference is insignificant. I mean, if you look at these numbers, say females 13 to 15 grams per deciliter, in males 14 to 16 grams per deciliter, these numbers overlap. So there's no significant difference over here. And sometimes people will say it is slightly higher in males than females. But if you look at these numbers, they overlap. And so we can say that there's really not a significant difference between the concentration of hemoglobin in males and females. It's almost the same. And you have known the function of hemoglobin to be responsible for the transport of oxygen from the lungs to the tissues, and then the transport of carbon dioxide from the tissues back to the lungs to be exhaled through the nostrils out of the body. So this is a basic thing which you have known. Now we are coming to build on this foundation that you already, have about hemoglobin. So let's move on. Let's go further to build on that foundation. We'll now continue to look at the structure of hemoglobin. The structure of hemoglobin. When you studied biological chemistry in first year, you talked about the structure of proteins, and you saw primary structure of proteins, secondary structure of proteins, tertiary structure of proteins, and quaternary structure of proteins. And I believe when you talked about quaternary structure of proteins, the example of hemoglobin was given to you. Now you see hemoglobin is made up of 4 subunits. So these are tertiary proteins that have come together in a complex, acting together as one molecule. You have 2 alpha subunits, alpha 1, alpha 2, and you have 2 beta subunits, beta 1, beta 2. Together, you have 4 subunits of proteins in the structure of hemoglobin. Because of that, you refer to the structure of hemoglobin as tetrameric. It's a tetrameric protein. And you also see at the center there. Now, these red squares are just depicting the prosthetic part of this molecule. That is the heme. So the heme is what I was talking about earlier as an allosteric part. And so you see, you can refer to the structure of hemoglobin as tetrameric allosteric protein. And the prosthetic part, that is the non-protein part of this molecule, is the heme. And the protein part, which is the alpha chains and the beta chains, that is those are the globins. And this part too sometimes is referred to as the apoprotein part. The same way as if you were talking about enzyme, you refer to the protein part as an apoprotein, which together with a non-protein part, a prosthetic part, which is a cofactor, a metal ion, or maybe even a coenzyme. It can be an organic compound like, things that like nicotinamide nucleotides or some vitamins, vitamin B, coenzyme A. All these things are organic compounds, but they are non-protein, which act in complex together with the protein part as prosthetic part. So this is similar in that case that you have, you have the apoprotein part, which is the globin, and then you have the prosthetic part, which is the heme. Now, when you were studying the primary structure of proteins, you refer to that primary structure as the sequence of amino acids in the protein chain. So if you take a protein, the protein is made up of amino acids. And those amino acids are bonded together. What's the bond that is between the amino acids? The peptide bond or amide bond, peptide linkage, however you wish to call it. Now, it means that you can actually take the structure of a protein and describe that primary structure to say this amino acid binds to this. So maybe you say phenylalanine binds to leucine, leucine binds to lysine, lysine binds to aspartic acid, the next asparagine, histidine, like that. You can count them and know the number of amino acids that are in a protein molecule. Now, in the hemoglobin molecule, the alpha chains, the alpha subunits, each of them has 141 amino acids, and there are two. The beta chains contain 146 amino acids. There are also two. So when you do the math and you put these numbers together, the hemoglobin molecule contains 5 means that we have. This structure that we are referring to, what we are looking at, is just one of the types of hemoglobins that we are referring to. Because this is the type of hemoglobin you see that we have other types of hemoglobin. It's only that those other types of hemoglobins are in lesser amounts in blood. So by a lot of times, when the structure of hemoglobin is being referred to, this is the structure that is talked about the most, having 2 alpha chains and 2 beta chains. Now, because you had studied proteins already, you have studied proteins, and you already know the structure of protein to say, well, protein has primary structure is containing a certain number of amino acids with peptide bonds between them, and these proteins have a secondary structure, the beta chain, the beta sheets, the alpha helix. You went on look at tertiary structure when you're having some bonds like hydrogen bonds that are between some amine groups and some carboxylic acid groups and things. You know all of those structures already. And so we're not going to focus so much on the the structure of the protein. That you already know. What you're going to do is to focus on the structure of the heme. So you are adding this knowledge to what you already know about proteins. So now you will know that this is the structure of the heme, in addition to the structure of proteins, which you already know. Now, before then, you want to know that the heme structure is mainly made up of what we refer to as pyrroles. So this is a pyrrole. What you see here on the left is a pyrrole, the ring, the five-sided ring with a nitrogen in there. So that's a nitrogenous ring you are seeing there, an azo ring you are seeing over there, an azo compound. It's a pyrrole. If you look at it, it looks, it has some resemblance with furans. If you did carbohydrates also in the first year, you saw you had some paranosis and furanosis. And the furan is what you are seeing here. This is kind of ring you see in sugars like fructose. You have oxygen in place of the nitrogen. And you can actually synthesize pyrroles from furans just by the process of amination of the furan. You have a pyrrole. Now you find these pyrroles porphyrins. And this is a structure you find in pigments like hemoglobin. And even later, you come to see that these porphyrins, which are found in pigments, it's not only in human beings, as we are talking about hemoglobin, that you find these porphyrin rings, these porphyrin structures as pigments in animals, say as hemoglobin, for instance. But you actually will find similar structures with plant pigments like chlorophyll. So we'll see that you see that these structures are actually very similar. So let's move on and we'll take a look at the structure of the heme. The structure of the heme. Now, if you pay close attention to this circular structure, this ring structure that you are seeing on the left, and you look at the letters A, B, C, and D. Now, each of those rings are pyrroles. So, the hemoglobin molecule, I said, is mainly of porphyrin rings. The porphyrin is made up of 4 pyrroles. So if you look at A, and you take a look at that nitrogen there, you see that nitrogen is in a 5-sided ring. So that is a pyrrole. You take a look at B, again look at the nitrogen. And you see the nitrogen there is also in a 5-sided ring. So that is the second pyrrole. C, you take a look at the nitrogen, and you see that nitrogen is also in a 5-sided ring. So that is the third pyrrole. And D, same thing, nitrogen is in a 5-sided ring, the fourth pyrrole. So 4 pyrroles are joined together by what we refer to as methynyl bridges. So between A and B, there is a linkage. That is what we refer to as a methynyl bridge. Between B and C is another methynyl bridge. And between C and D, another methanal bridge. And then the last, between D and A, there is that fourth methanal bridge. So pyrrole rings joined together by methanal bridges give you what we refer to as a porphyrin ring. So it's a porphyrin structure. It's a circular structure, a cyclic structure. And at the central position of that cyclic structure is ferrous ion, Fe2 plus, the ferrous ion. And the ferrous ion is linked to those nitrogens that are in the pyrrole rings. And you see here, it is actually the oxygen that is carried by hemoglobin, is carried on that ferrous ion. So you see it being depicted there. There is that, weak bond between some form of, will it be ionic bonding, weak between Fe2 plus and the oxygen molecule. Then you also see, something is depicted here. You have histidine of globin, which is to say, remember that this structure is non-protein. It is the prosthetic part. And it is where in each of the globin chains, each of the globin subunits. So the globin subunit, which is a protein, is made up of amino acids. And we said those amino acids, you can actually tell which position which amino acid, occupies. You have some histidines, some histidine residues that also the nitrogen in what you refer to as the imidazole ring, that is in the side chain of histidine, is interacting with that ferrous ion. The same way you have this Fe2 plus interacting with those nitrogens that are in the pyrroles. So, actually, when you go further, you study the structure of hemoglobin. I don't remember actually the particular positions of these histidine residues. I think one of them at the 92nd position, one is it 68, 53rd position, things like that. If you are interested, you can go and check more on those things. The histidines in the alpha chains and the beta chains, the particular positions that these histidines that interact with the ferrous ion at the center of hemoglobin. Those are there if you are studying much more into detail the structure of hemoglobin. Now, another thing is that on each of these pyrroles, the A, B, C, D pyrroles that we have looked at, you see that there are some letters that are written on those pyrroles. M, V, and there is M, P on some on C and D, you have MP. On A and B, you have MV. What do those mean? The M refers to methyl group. So that's these are carbon chains that are on the pyrroles that are in that structure. So you have methyl group that is depicted by M. V is a vinyl group. And P is a propional group. So that is what those letters stand for. So basically the structure of heme is what we have just looked at. We'll continue to look at the types of hemoglobin. I refer to the fact that there are some other types of hemoglobin. but we mostly refer to the HbA1 structure containing 2 alpha and 2 beta subunits. Because, as you can see here, when you look at the total hemoglobin in percentage in adult blood, it is 90% of all the hemoglobins that are in the blood. 90%. The HB1 composition, 2 alpha globins, 2 beta globins. You have also HbA2, which is having 2 alpha globins, 2 delta globins. Actually, we'll come to, as we continue, I think in the next lecture, we'll see what this alpha, beta, alpha, delta, where it's coming from. We'll come to see that it's actually coming from like different genes that are coding for the proteins that become the globin chains. That is where you get these different types of proteins coming from. And you see the HbA2 percentage less than 5. Now we also have fetal hemoglobin. Fetal hemoglobin. Bring your practical mind to it. The fetus in the uterus does not breathe oxygen from the atmosphere. It doesn't take in air through its nostrils and into the lungs. No, the fetus takes blood, takes the oxygen from the blood of the mother through the placenta. So it's a different type of hemoglobin that is working there. The placenta has a barrier. So you don't have the blood of the mother and the fetus just mixing up. No, there is a barrier between there. And there is that exchange of substances across the barrier. And the hemoglobin, which is in the fetus, that takes oxygen from the mother through the placenta, is the alpha, consists of the 2 alpha globins, 2 gamma globins. But you see, after birth, just over a period of, say, between 24, 48, 72 hours, after we are exposed to air, breathing in oxygen, the fetal hemoglobin gets converted to this HbA1 hemoglobin. Of course, there are some rare conditions in some individuals where they have persistent fetal hemoglobin that you have in such persons, still a significant amount of the fetal hemoglobin not converted to the HbA1 hemoglobin in such rare conditions. And they will have to be, for some time, be on oxygen. Some need some blood transfusions to deal with such issues over a certain period of time, things like that, because they have some genetic disorders that later they are not able to synthesize these enough of the beta globins, which is required in the HbA1 structure. Those are some rare conditions that occur. Then we also have the last one we're looking at here is what we refer to as glycated hemoglobin. Some amount of glucose that is in blood conjugates with hemoglobin. And of course, those of you working in clinical settings, for instance, you have been doing some diabetic tests, based on this, glycemic to analyze some glycemic conditions over a period of maybe some months, 3 months, for instance. Take an average to see if you have, the amount of glycated hemoglobin increased, signals, some condition, some diabetic condition. If it's within normal, then that's okay. But this also is in total, if you look at the amount of hemoglobin which is in blood, glycated hemoglobin is less than 5. It's less than 5% in blood. So these are, some major hemoglobin types that we have. Now, myoglobin. You remember we talked about the fact that there was a Nobel Prize that was won for, the discovery of the structure of hemoglobin, but together with myoglobin. Now we saw that hemoglobin was a tetrameric protein. That is, it's made up of 4 protein subunits. But myoglobin is a monomeric protein. So if just think of it as you have a hemoglobin, which is made up of 2 alpha chains, 2 beta chains. But myoglobin is just made up of 1 protein, globin with a heme in it. And that's it, nothing else. And that monomeric is a hemoprotein, consists of 153 amino acids. You'll find myoglobin in cardiac and skeletal muscles, because what myoglobin does is it serves as a reservoir and a carrier. It serves the function of an intermediate between the hemoglobin and the tissue cells, so that when hemoglobin brings oxygen from the lungs to the tissues, it transfers the oxygen molecule to myoglobin, and then myoglobin transfers to the cells. So that's the function that is serving. Hemoglobin transfers to myoglobin, myoglobin transfers to the cell. So that's that kind of intermediate between hemoglobin and the tissue cells, particularly for damaged tissues requiring rapid repair. Myoglobin is very active in these tissues, because oxygen has to be transferred quickly to these tissue cells for their metabolic processes that should quickly result into the repair of the damage that is in these tissues. Right. Now, let's compare the binding affinity of hemoglobin to, not affinity, what we're referring to is saturation. That is, how much of oxygen can hemoglobin and myoglobin take to become saturated. I mean, that is to say, if you are looking at these oxygen dissociative curves of myoglobin, which is red, and that of hemoglobin, which is blue. On a vertical axis, we have saturation with oxygen, percentage saturation with oxygen. And we have on the horizontal axis, partial pressure of oxygen in millimeter mercury. You can think of these as concentration of oxygen, but it's, because it's a gas, you're referring to it in terms of pressure. So partial pressure of oxygen. And you see we have myoglobin, which is monomeric, which means there is only one globin with the heme. And you see, when we saw the structure of the heme, it was the central ferrous ion that was carrying the oxygen molecule. What it means is that one heme, or if you think of it, one subunit of the hemoglobin molecule carries one molecule of oxygen, which means hemoglobin can carry 4 molecules of oxygen. Now, myoglobin takes only one, and then that is it. So if you look at these two curves, these two oxygen dissociative curves, you realize that at low partial pressure of oxygen, myoglobin has already gotten saturated. See the graph, it goes up like there's saturation on a vertical axis at such very low partial pressure of oxygen. Because it is only one molecule it carries, and that is it can't take any more. It's saturated. It cannot take any more. It gets saturated. That's it. But see hemoglobin, you see the saturation occurs in the lungs because it is in the lungs that hemoglobin takes oxygen. The air that we breathe in is rich in oxygen. So that is where the partial pressure of oxygen is high. And at the same time, hemoglobin is carrying 4 molecules, 4, which is 4 times more than what myoglobin can carry. And so you see that the partial pressure at which hemoglobin gets saturated is much, much higher. See where hemoglobin is getting saturated. Let's say somewhere where the curve is plateauing is above 50 millimeter mercury. That is where hemoglobin is getting saturated. But myoglobin, somewhere, let's say this is 10, just about 10 millimeter mercury, myoglobin is already getting saturated. And you see in the tissues where hemoglobin releases oxygen to the tissue cells, there you see the partial pressure of oxygen is lower. Of course, because you have the process of metabolism in the cells where they are making use of the oxygen and producing the byproduct carbon dioxide. So in the tissues, oxygen is lower in concentration. The partial pressure of oxygen in the tissues is lower, whilst carbon dioxide there will be higher. We'll come to see more about that. So you see the saturation there of hemoglobin is lower. So that is to compare the binding of oxygen to hemoglobin and myoglobin. You see when you pay attention to this curve of hemoglobin, you see it's gradual. It's gradual. And that too, we'll see why it is so. Why it's gradual. Why is it that the 4 molecules that do not just bind at once, and you see that it also follows the same trend as myoglobin, so that it gets saturated even at such low pressure. But it has to be at a high pressure that it gets saturated. To understand that, we look at what we refer to as the cooperative binding of oxygen to hemoglobin. In other words, the binding of oxygen to hemoglobin is cooperative. What does it mean that the binding of oxygen to hemoglobin is cooperative? Now, if you look at this mechanism, which is shown here at the top, you see this mechanism from left to right. Let's look at it from left to right. Now, from left, the first thing you see is 4 squares. Now, those 4 squares refer to the 4 subunits of hemoglobin. This is empty. There is no oxygen there. So that is a deoxyhemoglobin. There is no oxygen attached. No oxygen is bound on that hemoglobin. First step, an oxygen molecule binds to one of the subunits. That's what you see. An oxygen molecule binds to one of the subunits, and you have 3 of the subunits still unbound to oxygen. Now, what that does is that the moment one molecule of oxygen binds to one of the subunits, it increases the affinity of hemoglobin for oxygen. Then, a second oxygen molecule comes to bind to another subunit. So you now have 2 oxygen molecules bound on 2 of the subunits, and you still have 2 subunits that are unbound to oxygen. And that second molecule of oxygen that comes to bind also increases the affinity of hemoglobin the more for oxygen. So it goes in for the third oxygen molecule. So a third oxygen molecule comes to bind to a third subunit of the hemoglobin. And that continues until you have a saturated hemoglobin molecule, an oxyhemoglobin, fully saturated with 4 oxygen molecules. Now, what we have just described is what we are referring to as the cooperative binding. Cooperative in the sense that when one oxygen molecule binds, it increases the affinity of the hemoglobin molecule to go for more oxygen. And that affinity, that increase in affinity for oxygen continues as more oxygen molecules are binding until you have the hemoglobin molecule getting saturated. That is cooperativity. But what we have looked at is what happens in the lungs, because it is in the lungs that the hemoglobin is taking oxygen to take to the tissues. You see the arrows are pointing in 2 directions. Now, when we look at it in the opposite direction, that is from right to left, that will be the process that is occurring in the tissues. That is the release of oxygen molecules by hemoglobin to tissue cells. That is also cooperative. And you see that release, if you look at it from right to left, when first oxygen molecule is released, it reduces affinity of the hemoglobin molecule for oxygen so that it releases a second molecule. which reduces the affinity of hemoglobin the more for oxygen, and it releases the third until it releases the last one and becomes a deoxyhemoglobin. And that deoxyhemoglobin can bind to carbon dioxide to go back to the lungs, which can be exhaled out of the nostrils as a byproduct out of the body. Now we can talk about different forms of hemoglobin, because we've been talking about the fact that when oxygen binds, it increases affinity. When oxygen is released, it reduces affinity. Why that? So you have known that in the quaternary structure of proteins, you have different bonds there. Actually, those are bonds which you find also in tertiary structure, like hydrogen bonds, disulfide bridges, salt bridges, hydrophobic interactions. Those were bonds which you saw in tertiary structure of proteins, and it is those same bonds that you find between subunits that bring these subunits together to form a quaternary structure of proteins. Now, we have what we refer to as the T and R forms of hemoglobin. What do these mean? T refers to taut or tense. You can say tight, tightly bound together. And R refers to relaxed form of hemoglobin. Now, the T form of hemoglobin is a deoxyhemoglobin, hemoglobin with no oxygen bound to it. And you have the R form being hemoglobin which has oxygen bound to it. So that is oxyhemoglobin. You have oxyhemoglobin having the R form. You have the T form being deoxyhemoglobin. Now, what do you see? You see that between the alpha subunits, you have some weak ionic bonds. You have some hydrogen bonds that are between them, holding these subunits together. You also have some strong hydrophobic bonds between the alpha and beta chains. Now, you see that when oxygen binds, so if you pay close attention, these subunits that you see, at the center of each of these, the hemoglobin molecule is depicted with some red disc, and there is some, violet half, some half semicircle shown there. That is to depict that, like, that is the heme, the prosthetic part of the hemoglobin. And when you look at the right side, you see that on that heme, we have some blue structures. That is to depict oxygen bound onto the hemoglobin molecule. And you realize that the weak and the weak ionic and hydrogen bonds that you find between these alpha chains being shown here have reduced. When you look at the right side, there is that conformational change. So when oxygen binds to the hemoglobin molecules, it disrupts, that binding disrupts this bonding between the subunits, which causes that relaxation between the subunits. The molecule becomes relaxed, not tight together, not held tight together, not tense, not taut. That's why it's referred to as the relaxed form. And it is the molecule that is carrying oxygen, and so it's oxyhemoglobin. in that, in that form, you say the hemoglobin molecule has high affinity for oxygen, whilst in the third form, it has low affinity for oxygen. And this conformational change is reversible, as we saw with the cooperative binding and the cooperative release of oxygen. Right. Now, we can continue to look at the function of hemoglobin in the transport of oxygen. That mechanism of the transport of oxygen and carbon dioxide. How does hemoglobin do that? Now, on top here, if you are looking at this diagram, which is on the left, the top circle there is depicting the lungs. And the circle there is depicting in the tissues, what happens in the tissues. And we see that in the lungs, oxygen binds onto hemoglobin molecules. So that's a hemoglobin. You have a hemoglobin molecule, which is, showing here, and you see it's a tetrameric, and it's having the ferrous ion that's on which you have the oxygen molecules binding. So you have 4 oxygen molecules from the lungs that are binding on the hemoglobin. And in the tissues, you see oxygen is released by the hemoglobin molecule to myoglobin. And myoglobin will then release it to the tissue cells. So there is that intermediary function of myoglobin there occurring in the tissues. When it releases, it comes to peak again. It releases, it comes. So you have myoglobin molecules doing that work of an intermediary between the tissue cells and hemoglobin. And you also see that the oxygen molecules are used by the cells for cellular respiration, some metabolic processes. I mean, think of it as what are some of these processes you looked at when you were doing biological chemistry, like glycolysis, Krebs cycle. Those are processes to generate energy. You have 2 molecules of carbon dioxide shown here, bound on hemoglobin. That is a carbamyl hemoglobin. What is shown as green. And that carbon dioxide, carried on hemoglobin goes back in the lungs. Carbon dioxide is released by the hemoglobin molecule, and it is breathed out through the nostrils. Carbon dioxide being released out by hemoglobin carrying them back to the lungs caters for only 15% of the carbon dioxide which is produced as a byproduct in cellular respiration, which means it is left with 85% of carbon dioxide. What will happen to this 85% of carbon dioxide? We'll take a look at that. Of course, carbon dioxide, even though we are talking of it as a byproduct, when you actually look at biochemical processes, in a whole, not as, when we are studying it, we want to simplify it. So we take one system and study, we take another system. But if you begin to bring all the systems together, you begin to see that even a part of the carbon dioxide is also used in some synthetic reactions to produce some carbon dioxide proteins. Because you've come to find out that carbohydrates, proteins, lipids, nucleic acids, you see they are mainly made up of carbon, hydrogen, oxygen. Then you have nitrogenous compounds like proteins and nucleic acids that will be having nitrogen in addition. And some of those will have sulfur in them, and some will have phosphorus. So these are the major elements that you find in biomolecules. So of course, we talk of carbon dioxide as a byproduct, but some of it can actually go into some biosynthetic reactions. But let's take it that all of it is excreted out of the body. But still, 15%, and you are left with 85%. That's a lot. What will happen to that 85%? So let's move to that. What will happen to the rest of the carbon dioxide after just 15% of it is carried by hemoglobin to be exhaled out of the body? Now, the rest of that carbon dioxide undergoes the process that you are seeing now on your screen, transported as bicarbonate. Now, on the left-hand side, you see the process which is occurring in the tissues. The process occurring in the tissues. And on the right-hand side, we see the reverse of that process occurring in the tissues in the lungs. Now, what happens to the rest of the carbon dioxide? First, that carbon dioxide reacts with water to form carbonic acid. The enzyme which is acting in that process is carbonic anhydrase. The carbonic acid then dissociates into bicarbonate ions and hydrogen ions. Now, quickly, if you are having carbonic acid dissociating into bicarbonate ions and hydrogen ions, then it means that can result into decrease in pH in blood because you're having increase in concentration of hydrogen ions from your basic chemistry. So what then will happen? And here you see hemoglobin bringing oxygen molecules, releases the oxygen molecules to the tissue cells and binds the hydrogen ions. So what it means is that part of the hemoglobin, hemoglobin molecules carrying that carbon dioxide will also carry hydrogen ions as well. Then what happens to these bicarbonate ions? These bicarbonate ions are free flowing in blood, unbound. And hemoglobin back in the lungs. All this is happening with the bloodstream. So as hemoglobin reaches the lungs, bicarbonate ions in blood now recombine, they react back with the hydrogen ions to form carbonic acid. So that is the reverse of the process which was occurring in the tissues. And that carbonic acid dissociates into carbon dioxide and water. Then we breathe it out in our nostrils as carbon dioxide and water vapor. Hemoglobin molecule is free, picks 4 molecules of oxygen again, and goes back. So this is what happens to that large amount of carbon dioxide which is left. Is broken down into hydrogen ions, bicarbonate ions, and hydrogen ions bound to hemoglobin to take it back to the lungs. Bicarbonate ions free flowing in blood back in the lungs, bind to get back with the hydrogen ions to form the carbonic acid back, which dissociates into water and gets exhaled. Now we can talk about what we refer to as allosteric effectors. These are chemical entities. These are what we refer to allosteric effectors. They facilitate the release of oxygen from oxyhemoglobin, hemoglobin which is bringing oxygen from the lungs. When it gets to the tissues, there are factors that facilitate the release of the oxygen as fast as possible to the tissue cells. The first of it is partial pressure of carbon dioxide. In the tissues, we have carbon dioxide there in more concentration than oxygen, because there you have biochemical processes occurring, what we refer to as cellular respiration, which is producing carbon dioxide as a byproduct. And that high concentration of carbon dioxide serves as a facilitator for oxygen to be released from the hemoglobin molecules, so that quickly oxygen is released by hemoglobin and it picks the carbon dioxide molecules. Because the carbon dioxide is much more in concentration than the oxygen molecules there. It's just like, the way when you're looking at enzymes and you refer to competitive inhibition, where you have molecules competing for the active center of an enzyme. The more concentration of an inhibitor or the more concentration of a substrate to displace the substrate to displace the inhibitor, which is bound to the enzyme. Similar thing which is happening over here. You have carbon dioxide, which is higher in concentration than oxygen. Displaces that oxygen. Oxygen is released to the tissues, and carbon dioxide binds to the hemoglobin molecule to be taken back to the lungs to be exhaled. Now you also remember that part of that carbon dioxide reacted with water to give you carbonic acid, which dissociated into bicarbonate ions and hydrogen ions. And I quickly mentioned that it means pH will be reduced. You'll be having an acidic condition there. And so that high concentration of hydrogen ions is also an effector, facilitating so that oxyhemoglobin releases oxygen quickly and picks the hydrogen ions. It's a way of not having such an acidic condition becoming detrimental to the body. Now, remember that this cellular respiration, this metabolic process, which is producing this carbon dioxide that we are referring to, is happening in the cell. No particular, let's say the mitochondrion. That is where energy is produced. Which means when we say hydrogen ions being released, these hydrogen ions should be coming out of the cell to the extracellular space. That is into the plasma before you can have the red blood cell or specifically hemoglobin in the red blood cell picking these hydrogen ions. It must come out of the tissue cells. What it means is that if you have these hydrogen ions carrying positive charge, crossing the cell membrane, that's the tissue cells coming out of the cell, that will create a potential. It will create that electric potential across the cell membrane. That has to be compensated for because you are having positive charges coming in one direction. You will soon have more positive charges on one side of the cell membrane than the other. It will create a gradient, which means these positive charges will be flowing back again. So to compensate for that, there has to be some negative charges that will flow with it. And so you have chloride ions that are also moving together with the hydrogen ions to compensate for that positive charge so that it will compensate for the charges that are coming out of the cell. So you don't have that potential created across the cell membrane. In that case, you have chloride ions also facilitating the process of the release of oxygen from the hemoglobin molecules. Now, the last allosteric effector, 2,3-biphosphoglycerate. Where did you see these? You saw these in the metabolism of carbohydrates in the process of glycolysis. If you go and look at it, you see that this compound, 2,3-biphosphoglycerate, is an intermediate. The receiving glycolysis, you have some intermediate somewhere which is also having some function, even here. And you see, what does it do? Biphosphoglycerate is produced normally, in all red blood cells. You find 2,3-biphosphoglycerate binding to hemoglobin molecules. And it is to help with regulation, of the hemoglobin molecule having lesser affinity for oxygen. It's R form of hemoglobin. And I think those of you also working in clinical settings, if when you do some blood tests, for instance, for anemic conditions, you may notice that sometimes you have something written there, maybe 2,3 BP gene, or maybe you just say BP gene, biphosphoglycerate. It is because in anemic conditions or hypoxic conditions, as a form of adaptation of the body, that condition of lesser amount of oxygen for the cells, it produces more biphosphoglycerate that binds to hemoglobin molecules, so that the hemoglobin molecules, whichever amount of oxygen it is bringing from the lungs, can be released quickly to the tissues, because it's not the hemoglobin molecules are, they are lesser in amount as normal in anemic conditions. So biphosphoglycerate has to facilitate the release of oxygen faster. So you have these 2,3-biphosphoglycerate produce them more. So when you do the test and you have, elevated amounts of 2,3-biphosphoglycerate, it shows, some anemic condition. Another practical application of this is in athletes. So you remember sometimes when you have a sportsman, preparing for some big tournament. You hear sometimes maybe Ghana Black Stars has gone to Kenya to go to the mountains to go and train there. Why that? They talk about adaptation. Now, the scientific background of this, the biochemical background of this is that at such higher altitudes, when you go to a higher altitude, you are going to be at an altitude where you have lesser atmospheric pressure. And at a lesser atmospheric pressure, there's also lower amount of oxygen. Call it a hypoxic condition. So a lesser amount of oxygen will cause the body getting adapted to that condition to be producing more of these 2,3-biphosphoglycerates in the blood, so that there will be more of relaxed form of hemoglobin. So when you come back to a lower altitude where there is a higher atmospheric pressure, more oxygen, there will be a higher performance of the body, because the oxygen will be released faster. Even though it is more, it means it will be releasing oxygen faster as if it is still at that hypoxic condition, because it will take some time before the body comes back also to that condition of a higher concentration of oxygen that you have come to. And because of that, higher release of oxygen, the body performs better. So that is a biochemical, background of what, even in sports, you are having such an application there. Now, we can take a look at that effect of pH that we refer to, looking at the oxygen dissociation curve, what we refer to as the Bohr effect. We have seen this curve already with, the dependence of saturation of hemoglobin on the partial pressure of oxygen. And what you notice here is that you have the red curve is for a pH 7.6 and the blue curve for pH 7.2. What does it mean? I mean, pH 7.2 is lesser, it's more acidic than the pH 7.6. And you see there is that shift of the curve because there is a lower affinity of hemoglobin for oxygen at that lower pH value, which means you need more amount of oxygen for the hemoglobin molecule to be saturated. So you see the curve shifts to the right, and you see that to about maybe 50 millimeter mercury of saturation. But at pH 7.6, somewhere around 25 thereabout millimeter mercury, you are having the curve being saturated. So that is an effect that is referred to as the Bohr effect. Just to look at much more of what we referred to previously, the pH being an allosteric effector. Look at it, with this curve, and you see that 2,3-biphosphoglycerate. 2,3-biphosphoglycerate. Also, we talked of it as an allosteric effector. Two phosphate groups carrying negative charges, disrupting the ionic bonding that, the weak ionic bonding that was between the subunits of the hemoglobin molecule, making it relaxed. So you have the R form, which means the affinity of hemoglobin for oxygen there will be lower, because in the relaxed form, it will begin to, it will release the oxygen molecules faster. And you see the same thing, you will see a depiction of, say, the blue line. If you're looking at the oxygen dissociation curve, the blue line is normal hemoglobin that is having a 2,3-biphosphoglycerates, so in normal blood. And if you have, for instance, 2,3-biphosphate removed, if you have lesser amount of it, what you see is that you have the affinity of hemoglobin increasing. Because of that, you have saturation of the hemoglobin molecule occurring at a lower partial pressure of oxygen. The opposite happens in anemic conditions, where you have higher amounts of 2,3-biphosphoglycerate. You see, you need a higher amount of oxygen for hemoglobin to be saturated. In other words, the hemoglobin molecule has lesser affinity for oxygen, which means it will be releasing the oxygen molecules faster to the tissues. So that's what you see there. Now we can end with some two major derivatives of hemoglobin that are referred to in some health conditions, what we refer to as methemoglobin. That is the oxidation of the ferrous ion is Fe2 plus being converted to Fe3 plus. That's an oxidation reaction which can occur, by the action of oxidants like hydrogen peroxide, some free radicals, even some drugs. Like there are some drugs like some antibiotics like sulfonamides, for instance, can cause, some of these oxidation of the ferrous ion to ferric ion. Some local anesthetics can also do this. But the good thing is, even naturally without these drugs, you have some amount of the ferrous ion getting oxidized to ferric ion. But there is that quick conversion. The body has its own mechanism. With the methemoglobin reductase, that enzyme that reduces the ion back to the ferrous ion, so that it must be in some really severe conditions that you'll be having methemoglobinemia. Means that maybe some consumption of a substance high in concentration, or some kind of an acute exposure to some chemical may result into such condition as methemoglobinemia. Then we can talk lastly about carboxyhemoglobin. That is when you have carbon monoxide binding to the hemoglobin molecule. In fact, carbon monoxide has been known to have 200 times more affinity to the hemoglobin molecule. Even though the hemoglobin molecule itself has some protective mechanism against inhaled and binds to the hemoglobin molecule. What it means is that if you are having carbon monoxide binding to, having such 200 times more affinity, when it binds to hemoglobin, it's difficult for the carbon monoxide to be released. And what happens? It means not enough oxygen is going to be carried to the tissues. It will start, causing some condition of call it cellular suffocation, and that will result into headache, nausea, vomiting, breathlessness. I think for our university, which is at the center of the city, where we are close to Agbogboshi, for instance, where every time they are burning things and we have engines moving around us. I think just beside us, even we have how do you call it? The Ghana Publishing Company there. That is, they have a chimney and you see always smoke coming out of it and things like that. You as scientists in the health sector studying some of these things, think you can be concerned about some of these things. And when you talk about it, maybe you can advocate for some better systems. We make a lot of noise, there's a lot of noise which is being made about people destroying forests, looking for some metals and things like that somewhere, and we seem not to see what is happening at the center of the city, which is the pollution there among a larger amount of people. That is equally as dangerous as what is happening even in those forests. But it appears not much of attention is paid on to these things. So as younger generations coming, I think maybe you can pay more attention to some of these things. I think I have talked and talked and talked enough. So I will stop here. And now listen to you. So I'll keep quiet. It's time for you. You can write in the chat, or you can raise your hand and I will enable your microphone. You unmute yourself and you speak. The floor is open. Now, as you are preparing, thinking of